Good evening, everyone. This is your week 2 problem solving session, and it will be similar as that of week 1. So, in this also, we will firstly talk about the week 2 topics, we'll summarize them, and then we'll practice few MCQs. Along with that, we'll also look into the current literature, that what is going on these days. And if you have any doubts regarding the assignments, then you can ask them. So, in week 2, it was majorly about the amino acids, their properties, and then about the structures of proteins, how they fold. So all those topics will be covered, initially starting with the amino acids and their properties. So as we all know that these amino acids are the building blocks of the proteins. So you can also say that these amino acids are like forming the polymers, and then it gives rise to protein. And each of them, they have unique chemical structure, which is determined by the R groups. So this is the general composition of amino acid. These are the groups which are present in an amino acid. So one in the middle there is an alpha carbon which is linked to the carboxyl group which contains COOH, and then this alpha carbon is linked to the amino group which is NH2. On the one side it has hydrogen and on the other side it has R group which is the side chain, and in case of glycine it is hydrogen, otherwise for other amino acids it is some side chains which are present, and this is the group which decides that what will be the amino acid, because it is the only group which varies among different amino acids, and these amino group they are present towards the N terminus. So as in the case of DNA and RNA, we have seen that the directionality is from 5 prime to 3 prime. So similarly in proteins the direction is from N terminus towards C terminus. So you will find that the amino group will be present towards the N terminus and carboxyl group will be present towards the C terminus. Then we have hydrogen atom and the R group. And these amino acids, they can exist in both the non-ionic as well as ionic forms. So non-ionic is the form in which there is no charge present on them. Or you can say when there is not positive or negative charge present on them. And they can also exist in ionic form, which means they can have uh, positive and negative ionic forms. And it depends that whether net charge is zero or whether it is positive or negative. So zwitterion is a ionic form in which net charge is zero. Although these groups even means individually they have some charge, but they cancel out the effect of each other. That's why they exist as the zwitterion form. And in the later slides, we will look into different amino acids and understand that in which form they exist. So now we'll talk about different types of amino acids. So there are 20 standard amino acids, although there are certain other amino acids also present which are produced as a result of metabolism, but they are not standard amino acids. So on the standard basis, we only discuss 20 amino acids, which makes up the proteins in living organisms. And these amino acids, they influence the chemical behavior, interactions, and role of protein structure and function. So as they have different R groups, so these R groups can decide that how this protein will interact, because we'll see that there are different constituents of these R groups. So different functional groups will be present, so they can determine that what will be the interactions, and if different interactions are present, they will determine that how this protein will fold. So in that way, they govern that what will be the function of a protein. So these 20 amino acids, they can be categorized into different types on the basis of the side chain, or you can say the R group. So they can be non-polar or hydrophobic. So as the name suggests, hydrophobic, that means which repels water. So water hating molecules, they are non-polar. So they will have certain groups which repels the water. Then there are amino acids which are polar or hydrophilic in nature. So these are the amino acids which interacts with water. Then comes acidic amino acids. So acidic amino acids are the ones which are negatively charged, and they have the negative charge because of the carboxyl group which is present. Then there are certain basic amino acids which contain positive charge, and this positive charge is given by the amino group. So they have additional amino group, and in this case they have additional carboxyl group other than the normally amino and carboxyl group which are present in the amino acid. So they're additional groups. Then comes aromatic amino acids. So they have aromatic rings which are present in the R group. Then there are sulfur containing amino acids which contain sulfur in their side chain. So in this way, these 20 amino acids, they are the building blocks of the proteins, because they will be arranged in different combinations, and in that way they means vary among different proteins. And that's why they bring about different functionality as well as different structure in different proteins. So now one by one, we'll look into different categories. So starting with the non-polar or hydrophobic amino acids. So these amino acids have side chains which are hydrophobic or water repelling. And means how this water repelling nature comes because they have hydrocarbons in their side chain. So these hydrocarbons, they are water repelling or non-polar in nature. So the characteristics of these non-polar amino acids are that they are found in the interior of proteins, because we know that the proteins are present inside the cell, and our cell is composed of means it's maximally composed of the water. So if proteins contain these non-polar amino acids, then they will be present interior of the protein, because protein is present in the environment which is aqueous. So to avoid that interaction, they are generally present towards interior of the proteins, or we can say these amino acid constructs the protein core, the core region of the protein, and they also contribute to protein folding by stabilizing the structure by hydrophobic interactions. So even in the previous session also we have seen that there are different forces which are responsible for the stability of the protein, tertiary structure or different structures of the protein. So these hydrophobic interactions, they are also responsible for stabilizing the protein structure and hence promoting the folding of a protein. So these are the examples of non-polar amino acids, starting with glycine, alanine, valine, leucine, isoleucine, methionine, tryptophan, phenylalanine, and proline. So these all comes under non-polar amino acids and they are hydrophobic in nature. So the components which are highlighted in blue, these are the R chains or R groups, and this is how they vary from one another. And one more thing which I want to add, that tryptophan and phenylalanine, so they contain the aromatic side chain, but they comes under non-polar amino acid. So means they can come under non-polar amino acid also and aromatic group containing amino acid as well, depending on the nature, that they are also hydrophobic in nature. That's why they are written here as well. Then comes polar or hydrophilic amino acids. So these are water loving. That means they have certain groups which can interact with the water molecule, which can form hydrogen bonds. So I hope you are aware of the hydrogen bonds. So these bonds are formed between the hydrogen, which is linked to the most electronegative element, to the another hydrogen, which is already attached to some electronegative element. So in this way, they will have the hydrogen group which is present and which is attached to the electronegative group or electronegative atom. So that's why they are able to form hydrogen bonds with the water molecules which are present. So what are the characteristics of these amino acids? They are found on the surface of proteins because protein is present in the aqueous environment. So these amino acids can interact with water. That's why they are present on the surface of protein and they interact with this environment. And then hydrogen bonding helps for the protein stability and interaction. And as you are, means I hope you are aware that in secondary structures, there is the hydrogen bonding, which is responsible for that secondary structure, which we see. So in that way, it is also helping in stability of the protein as well as its interactions with other amino acids. So these are the examples of polar amino acids such as serine. So in this case, you can see we have the OH, terminal OH group in the R. So this OH group is responsible for the hydrogen bonding. Then in threonine, we also have OH group along with the methyl group which is also present. So these are the side chain. In cysteine, we have sulfur group SH. So S is also highly electronegative. So that's why it will also be involved in the, so it is involved in actually in the formation of the disulfide bonds because it contains sulfur. Then we have tyrosine. So this is also an aromatic amino acid, but it has OH group. So that's why it has hydrophilic nature. We have asparagine as well as glutamine. So these also contain double bond O and NH2 group. So as nitrogen is electronegative and hydrogen is attached, so this NH2 group will be involved in the hydrogen bond formation. So these are the examples of polar amino acids. Now we'll talk about the acidic amino acids. So acidic amino acid contain carboxyl group. So when we say acidic nature, so acidic nature means that they can release the proton. So if carboxyl group is present, then it can release H positive ion and become negatively charged. So uh, means which makes them negatively charged because of the loss of this proton at physiological pH. And the characteristic of these acidic amino acid is that they are involved in ionic interactions because they exist in the charged form. So there will be charge-charge interactions, means two charged amino acids can interact with one another. That is referred to as ionic interactions. And they are found on the surface of proteins. And they interact with water or positively charged residues. So due to these charged or this nature, they can interact with water as well as other positively charged amino acids. So these acidic amino acids are aspartic acid as well as glutamic acid. And these aspartic and glutamic acid are also called as aspartate or glutamate. So these are the alternate names for them. And they contain COO, means this COOH terminal carboxyl group, which is involved in interaction. So in physiological pH, it is exist in the charge, negatively charged form because it loses H positive ion. That's why it is represented as C double bond O negative in both the cases, and they both vary by a single methyl group. So an additional methyl group is present in glutamic acid. And now we'll talk about the basic amino acids. So in acidic amino acids, we have seen that they have additional COOH group. Similarly, in basic amino acids, we have amino group, which is NH2, additional amino group in the side chain. So which can accept the H positive ion. That's why they act as basic, they are basic in nature. So they are basic amino acids, making them positively charged at physiological pH, because in physiological pH, they gain the H positive ion. So their characteristics are they are hydrophilic in nature and they are involved in salt bridges or enzyme active sites. So salt bridge is also known as the ionic bond, which is formed between two charged species. So in this case, negatively and positively charged species, when they interact with one another, they will form the ionic bond or salt bridge. So these charged species will be, means opposite, they have opposite charges. That's why they're interacting, because if they have similar charge, they will repel each other. So for ionic bond, both species should have variable charges or opposite charges. So that's why they're involved in the salt bridge formation. And along with this, their positive charge also plays a role in the stabilizing of protein structures. So in the later slides, when we'll see the how the protein folds and how it is, means takes different structures, then we'll see there are different interactions which are involved. So these interactions are one among them, these ionic interactions, which are responsible for the protein structure stability. So these basic amino acids are lysine, arginine, and histidine. So as you can see in the terminal position, they have this NH3 positive group. So amino group is present, which is responsible for the positive charge on them. And this is the difference, means how they vary from one another based on the R chain. Now the next type is aromatic amino acids. So when we are talking about different types, then don't confuse that these types are not independent types. So you might see these aromatic amino acids in the polar as well as non-polar groups also. So on the basis of nature, we have categorized them into different types and they can reoccur in other types as well. So when we say aromatic amino acids, they contain aromatic rings in their side chain, which can participate in hydrophobic interactions and pi-pi stacking in the protein structure. So whenever something has aromatic rings, so there are possibility of pi-pi stacking. And along with this, there can be hydrophobic interactions as well. So as there are hydrophobic and pi-pi stacking, so they are often found in the interior of proteins. And the other thing is that these, because they have aromatic rings, so they are slightly large in structure. So due to their larger size also, they are means more favored at the interior of the protein instead of means at protein surface. So they can participate in aromatic stacking interactions with within the proteins or the ligands. So these are some of the interactions which happen, which are also responsible for determining the protein structure and its stability. So aromatic amino acids are phenylalanine. So it's named as phenylalanine because its structure is same as alanine, but additional phenyl ring is present. That's why it is called phenylalanine. Then we have tyrosine, which contains this benzene ring attached to OH group. So it is generally polar in nature because it has OH group. Then comes tryptophan. So among all the amino acids, this one is the biggest, or you can say great, means big in size than other amino acids. So it is means less frequently present in the proteins because there will be many steric clashes if it is present. So it means its frequency is less as compared to other amino acids, and it has higher molecular weight as well, and it contains these rings. So these are the aromatic amino acids. Then there are sulfur containing amino acids because they contain sulfur, so that's why they are involved in disulfide bond formation. And along with this, due to these interactions, so disulfide bonds are the covalent bonds, which are strong interactions. So they help to stabilize the protein structures. And the characteristics are that cysteine, it is involved in disulfide bridge formation between the proteins. And when there is another amino acid, which is methionine, which is also a sulfur containing, but it is not involved in disulfide bridge formation. So they are involved in the protein synthesis as first amino acid in many polypeptides. So in different organisms, the methionine is the first amino acid. You can say that it is the start codon, which is AUG, so that codes for methionine amino acid. And these are the structures of cysteine and methionine. So in cysteine, we have terminal SH group which is present. So that's why they are polar in nature. They can interact with water molecules because they have SH group, and they can also form disulfide bond. Whereas in methionine, that sulfur is not exposed. It is present before the methyl group. So due to which they cannot directly form disulfide bonds, but means they have different interactions, and it is non-polar because it has terminal methyl group. So they are, due to which they cannot interact with the water molecules. So these are certain types. These are the types of different types of amino acid based on the constituents, that what are the constituents that are present and how they will interact. Then on the basis of their, means how they are synthesized, they are divided as essential versus non-essential. So essential means which are required, which means which we need. So that means they cannot be synthesized by the body, and they must be obtained from diet. So they are obtained from external sources. That's why they are essential. For example, leucine, valine, isoleucine, lysine, methionine, phenylalanine, threonine, tryptophan, histidine. So these are the amino acids which are not synthesized by our body, and we need them from our diet. Whereas non-essential amino acid means which our body can synthesize. So we do not need external supply of these amino acids such as alanine, glutamine, asparagine, serine, and glycine. So they can be produced as a result of other means reactions which are going inside our body. So we do not have to supplement them. And this is the list of essential versus non-essential amino acids. And the remaining amino acids, they are conditionally non-essential. So conditionally non-essential means that normally they are non-essential amino acids, but in certain conditions, our body might demand them in higher concentration than in the concentration which they are produced in. So in such situations, such as stress, illness, injury, or during the rapid growth, we require more of these amino acids. So in that condition, they become essential. We need to supply them from our diet. So that's why they are called non-essential, means conditionally non-essential. And this is the list, similar list for essential and non-essential amino acids. And in essential amino acids also, these leucine, valine, and isoleucine, they are the branched chain amino acids, and they makes up 35% of essential amino acids in our muscles. And these are different vegan sources which provide these amino acids. So this is just for your idea. Now we'll look into the ionic form of amino acids. But before moving on, so is everything clear till now? Do you have any doubts in the types of amino acids? Okay, thank you. Yeah, so now we'll talk about the ionic forms of amino acids. So we have seen that these amino acids, they can exist in the deuterion form, in which the net charge on them is zero. And they can also exist in the cation, that means which contain positive charge, as well as anion, which contains negative charge. And then there is a pH which determines that in which form the amino acid will be present. Either it will be present as the cationic form or anionic form. And these pK1 and pK2, they represents the equilibrium constants. So in the later slides, we'll see that what it means. So what happens in low pH conditions? Low pH means there are acidic conditions in which the molecule or the amino acid, it will exist in the cationic form. So how it exists in cationic form? Because in acidic condition, there will be higher H positive concentration in the medium or in the aqueous environment. So if higher H positive concentration is there, then it will lead to the protonation of amino acid. Protonation means that amino acid will gain that H positive ion. So if H positive ion is gained, then it is gained by the NH2 group. So NH2 or the amino group will become positively charged, whereas carboxyl group, so it already have the hydrogen, so it will not gain that H positive ion, so it will remain neutral in the low pH condition. So net positive charge on the amino acid will be positive. So in this way, at low pH condition, amino acid exists in their cationic form. And if there is high pH, that means the basic condition is there, or the amino acid will exist in its anionic form. So in basic condition, there is low H positive concentration in the medium. So if there is low H positive concentration, then the amino acid will undergo deprotonation. That means it will have a tendency to lose that H positive ion, because H positive ion concentration in the external environment is low. So in that conditions, the amino group, which is NH3 positive, it will convert to NH2, which will be neutral because it will lose the H positive ion. And the carboxyl group, which is COOH, So it will also loses the H positive ion because it also contains the hydrogen. So it will be COO negative, which is negatively charged. So now net charge on the molecule will be minus. So it will be a negative charge. So that's why the amino acid will exist as the anion in the higher pH conditions. So these pH conditions will determine that what will be the charge on an amino acid. And along with this, the R group will also determine because it also contain different constituents. So at neutral pH, which is around 7.4, these amino acids, they exist in zwitterion form. So zwitterion is a condition where the net charge is zero and both NH3 positive and COO negative, both the ions are present, due to which they cancel out the effect of each other, and that is a dipolar ion. So zwitterionic form is also known as dipolar ion form. Due to this nature, the amino acids means they act as the amphoteric species. That means they can exist as both bases as well as acids. So depending on what is the pH, they can act as acid also and they can act as base also. And this property is arised from the NH2 group. So because NH2 group has a tendency to gain H positive ion, so in those situations they will gain H positive ion, and the carboxyl group which contains COOH, so it has tendency to lose H positive ion. So in those scenarios, they will act as the H positive ion donating molecules. So these are the functional groups which provide them the ability to act as both acid and base. Okay, so in this, the similar thing is shown that NH2 is basic in nature. It can gain H positive ion and convert to NH3 positive, whereas COOH contains the OH, which is acidic in nature, so it will lose H positive ion and become O negative. And this amphoteric nature is also essential in protein structures, enzyme functions, as well as buffering capacity. So they act as natural buffers in biological systems. They try to maintain the pH, so they act as buffers. And when we talk about their acidic behavior, so acidic behavior means their proton donor nature. So in basic conditions, when the pH is high, they act as acid. So means this is to show you that how they act as buffers. So when pH is high, then they will means carboxyl group will be present, which will donate H positive ion, and they become carboxylate. This will make the amino acid act as an acid, where it is donating the H positive ion. And when the pH is low, that means when there are acidic conditions, then these amino acids will act as base. They will gain the H positive ions and convert into the ammonium ion, which is NH3 positive. So in this way, they act as base. So they are trying to buffer the solution. So they are acting as a buffering agents. Then comes the concept of isoelectric point. So now we have seen that they can exist as cationic forms, anionic forms, as well as zwitterion form. So isoelectric point is the pH at which these amino acids they are exist as zwitterionic form. That means there is no net charge on them, and this is governed by the side chain, because side chain is the one which vary among different amino acids. So due to this side chain, different amino acids can have different pi values. So pi value, to calculate pi value, what is the formula? So it is means equilibrium constant of COOH plus equilibrium constant of NH3 divided by 2. So we generally sum these equilibrium constants and divide them by 2, and we get the pi, which is the pH at which the amino acid it exists in the zwitterionic form. So we'll see a few examples that how it works. But before moving on to that, we'll clear our concepts about these. So as we know that now I'll talk about what equilibrium constant means. So when we talk about water, it is the molecule which can ionize. So it is ionizable. It can exist in the form of means in positive charge it exists as hydronium ion form, and in negative it is OH negative group. So equilibrium constant is calculated by dividing means dividing the concentration of H positive ion and OH negative ion with the concentration of the complete means joint molecule, which is H2O. So it is means equilibrium constant is the concentration of ions divided by the concentration of entire molecule. So according to Henderson-Hasselbalch equation, this is for the water, and for the any other molecule. For example, in this case, we are talking about the amino group as well as the carboxyl group. So in those situations also, the entire amino group will be present as a complex, and its ions will be taken as H positive and the other, which is means depending on the type of group which is present. So in that way, we will calculate the equilibrium constant. So which will be the concentration of ions divided by the concentration of the entire molecule. So this is just for your reference that at what pH the value of what is the value of H positive ions and what is the value of OH negative ions. So pH is the negative log of this equilibrium constant. So this is the link between the equilibrium constant and the pH. So pH is the sum of pK plus log of A negative ions divided by HA. So concentration of the entire complex. And now this is to show you that means when the pK value is equals to pH. So pH will become equals to pK when the concentration of A negative ion to HA ions means log of concentration of A negative to HA means the entire complex when that is 0. So at that point pH will become equals to pKa. And if this means ratio is less than 0, then that means pH is less than pKa. And if this concentration is greater than 0, then that means pH is greater than pKa. So one thing to see from here is that when the concentration of ions are higher than the complex, that means there are greater ions that are formed. So that happens in the condition when pH is high. And similarly, in this case, when the A negative ion concentration will be low, that means this ratio will be less than 0. In that condition, pH is less than pKa. So this is the link between pH and pKa. So this is just to show you that why we are taking means pKNH2 and pKCOH to find isoelectric point. So these all are telling us about the pH values depending on the ionic concentrations. So now we'll follow some examples to calculate the isoelectric point to see that how the side chain or the R group influences the isoelectric point. So simple example is of alanine, which is non-polar in nature. So in alanine, at physiological pH it exists as the charged means net charge is 0, deuterionic form. So CH and NH3 positive, COO negative and CH3. So when it gains H positive ions, so it means at pH less than 2.4, it has the NH3 positive state, and at pH greater than 9.8, it has means it loses that H positive ion. It exists in COO negative. Now we have to find this pH at which it exists in this form, in deuterionic form. So it will be pK1 plus pK2 divided by 2. So pK1 will be this one, that is the in which the NH3 positive ions are formed. So this is the value of pK1, which is 2.34, and plus pK2. So means pK of COOH, that how those COOH is broken down into COO negative and H positive. So this is the equilibrium constant. So its value is 9.69. So we will sum these values and divide them by 2. So that Pi value comes out to be 6.015, which means that at pH of 6.015, this particular alanine, it exists in the zwitterion form. So in the previous example, we have seen that at pH of near about 7.4, that is the physiological pH in which the amino acid exists as deuterion form. So that is the condition when there is means for the glycine that can apply. But in the case of this alanine, because it has the in the R group, it has methyl group. So due to which it's means that pH value is 6.015, at which it exists in the deuterionic form. Now we'll see for the basic amino acids, because they have additional NH2 groups. So how isoelectric value changes and to which direction it moves. So that also you'll see. So in this scenario, this is the deuterionic form when net charge is 0. So it has different equilibrium constants because it has additional means NH2 group which is present. So it has that tendency to exist means net, it can exist in the form where net charge over it is +2. So in this situation, it is existing in the NH3 positive form. So now see carefully that it has this N group. So it is gaining the H positive ion and it is existing as NH positive. Now the another gain of okay, so now in this this loses the H positive. The next gain of H positive is happening at this COO negative group. So firstly, this nitrogen which is present in this particular ring-like structure, so that H positive gain is present. Now it also contains CO negative group, so this has also the ability to gain H positive ion, so it can also exist in +2 charge state, where this has also gained the electron, means H positive ion, and this has also gained the H positive ion. And then it can also exist in -1, means it can have the net charge of -1 when this. So in this state, the NH3 which was present, it has lost one H positive ion and it becomes NH2. So previously this positive and negative were cancelling out the effect of each other, but now at means this dissociation, or you can say this equilibrium constant, where NH3 has broken down into NH2 plus releasing H positive ion, so it is in NH2 form, so the net charge is -1. So now in this case, how we'll find Pi? So we will sum up these pK values, this pK2 and pKr. We will not focus on pK1, so we'll focus on the adjacent pK values to the zwitterionic form. So we will sum up pKr plus pK2 and divide it by 2 to find the Pi values. So according to this plot, pKr is 6 and pK2 is 9.17. So they are summed and divided by 2. So in this case, the Pi is 7.585. So as in the polar, known polar amino acids we have seen, the Pi value was 6.015, which was less than the physiological pH value. Whereas in this case, the Pi value is 7.585, which is slightly higher than the physiological pH, which is near about 7 to 7.4. So which means that this R group is also determining that at which pH the amino acid will act as the zwitterion. And the another example is of glutamate, which is the acidic amino acid. So it contains the additional CO negative group. So it can exist in -2 form in addition to -1. So it can also have -2 charge as well. So in this case also, similarly to that of the basic amino acid, in acidic amino acid also, this is the zwitterionic form where net charge is 0. So in this also we will add pK1 and pKr, which are the adjacent equilibrium constants, and divide it with 2. So looking at the plot, pK1 value is 2.19 and pKr value is 4.25. So we will add them, divide by 2, and Pi comes out to be 3.22. So now here you can see that as we move towards the acidic amino acids, the Pi value tends to decrease. That means the acidic amino acids, they exist in zwitterionic form at the lower pH, whereas the basic amino acids, they can exist in the zwitterionic form at the higher pH, whereas the non-polar amino acids, so they are existing at the means zwitterionic form in the near to physiological pH, so nearly the same, not the higher but a little lesser, but near about 7 only. So this is all about the Pi value calculation and how the side chain is influencing the uh, isoelectric point calculation. So is it clear till now? Okay, now let's move to the next. So now we'll talk about the physicochemical property. Yes. Yeah, so pH is normally the what is the current power of hydrogen. So pH is power of hydrogen, but Pi is the pH at which the amino acid is existing in the zwitterionic form. So let me show you. So in pH can be anything. So pH is depending on the what is the means environment of that protein. So protein can exist in any form, means amino acid can exist in any form at different pH values. So pH means simply you are looking at the concentration of H positive ion in that particular medium. But when we are looking at the amino acid in the zwitterionic form and then finding the pH at which it is existing in that particular form, that is known as Pi. So it is the point. It is telling you the pH value at which the amino acid is in zwitterion form. And if you simply talk about pH, then amino acid can also be present in anionic as well as cationic, any form it can present at any pH. So Pi is specific to zwitterionic form. Is it clear? Yeah. So now we'll talk about the physicochemical properties of amino acids. So physicochemical properties means that what is the polarity, either it is polar, non-polar, what is the charge, whether it is acidic amino acid or basic amino acid, hydrophobicity. So about those non-polar amino acids which contain the alkyl groups, then aromaticity, then sulfur content. So these all are some physicochemical properties of amino acids which are responsible to determine that how this protein will fold, how different interactions will happen, because different groups which are present, they will determine that what kind of interactions are possible. So in this way, these physicochemicals are very properties are very important to understand, and these properties are also determining the conformation of proteins as well as their functionality. So this is a table which tells you some additional properties as well, such as the molecular mass, number of atoms, its volume, as well as hydropathy index. So hydropathy index are also the means this is a scale which tells you that whether the amino acid is hydrophobic or whether it is hydrophilic. So hydropathy index value is ranges from negative to positive. So if it is negative, that means it is hydrophilic in nature. That means it interacts with water. If it is positive, then that means the molecule is hydrophobic. So this is the means the range in which the values can be present. And in this table, means I have highlighted tryptophan, which is aromatic in nature, and this is the only amino acid in the entire amino acids which has the highest molecular mass, that is 204 daltons, and with means greater number of atoms also which are present, as well as its volume is also high. That means it requires a larger space. If it is present in protein, then greater space is required, due to which there are possibility of steric clashes and other things. So, and its hydropathy index is minus 0.9. Then the another amino acids. So in this case, I have highlighted glycine because it is non-polar aliphatic and it has the molecular mass of 75 daltons with least number of atoms, that is 10, which is present, and it is achiral in nature because it has two hydrogen groups, due to which the alpha carbon has two similar groups. So that's why it is achiral in nature. And this is the smallest amino acid among all. And due to the two hydrogen groups, due to its achiral nature, it is means it can exist anywhere, it can exist in any conformation because there are no steric clashes. So if there are bulky R groups, then there will be some steric hindrances, because if molecule will be large, if it occupies larger volume, so if two larger volume occupying R groups will be present, they will repel each other. But in case of glycine, we have hydrogen in place of R groups, so it does not, I mean, you can say it does not repel other molecules. So it is most favorable at different angles, and this also can occupy different, means it allows the molecule to rotate in any directions. So that is the means different physiochemical property of glycine from another amino acids. Okay, so now as we have talked about chirality in amino acids, which is determined by the alpha carbon, and glycine is an exception because it is achiral. So these amino acids, they can exist in two stereoisomer forms. One is L amino acid, another is D amino acids. And these L and D forms, these are the stereoisomers and they appear like a mirror image of each other. For example, if we talk about our left hand and right hand, so they are also the mirror images. But if we try to superimpose them, then they won't superimpose. They are non-superimposable. And in nature, the L amino acids are standard amino acids. So throughout evolution, we have seen that L form of amino acids are more favored than the D amino acid forms. So later on, we'll see that in which situations this D amino acids exist. So they are very means rare where we find the D form of amino acids. This is just to show you that how the L and D forms exist. Now we'll talk about the chirality affecting bacterial cell walls. So bacterial cell walls, so there are different two main types of bacteria, gram positive and gram negative. So in both of them, they contain NAG/NAM polymers. So the chirality effect, it is crucial to determine the structural integrity and functionality of these bacterial cell wall, particularly in the composition of peptidoglycan, because their cell wall contains larger content of peptidoglycan which is present. So peptidoglycan means that peptides which also contain sugar moiety attached to them. So these NAG and NAM, these are the sugar moieties, and these are the peptides. So peptide means when there is a short stretch of amino acid which is present, that is referred to as peptide. And if it is the larger peptide, means it means the length of peptide is large, so that is referred to as protein. So peptide means shorter amino acid content. So in this case, in NAG and NAM, they are attached to this pentapeptides. So these there are five amino acids which are present, so which exist in the L and D form. So both these forms they exist. And in case of gram positive, we have MDAP in place of L-lysine, which is in the gram negative form. This is the only difference among gram-positive and gram-negative bacterial peptidoglycan. Otherwise, its structure is similar. So in this case, we have the chain of NAG and NAM, which are interconnected to the another chain by these pentapeptides. So for this interconnections to take place, there is enzyme which is present, which uh, cross-link different NAG-NAM chains. So this, when this cross-linking happens, then there is loss of one of the D-alanine, which is present in the terminal. So if in the structure, if you see this cross-linked structure, you'll find that there are tetrapeptides which are present, and then there is a pentaglycine cross-link. So these are linked by the pentaglycine. So means five glycine amino acids will be present, which will be linking them. So link bond will be formed between D-alanine of one of the means NAG, which is attached to this tetrapeptide. So this D-alanine is linking to the another L-lysine, which is present in the another NAG, which contains this tetrapeptide. So this cross-linking is happening by the D-alanine, and one D-alanine is lost during this reaction. That's why we see four instead of five in the peptidoglycan structure. So the sugar components are NAG, which is N-acetylglucosamine, and NAM means N-acetylmuramic acid. And these are also the chiral molecules, and they exist in two mirror image forms, that is L and D forms. But specific forms are only biologically active and functional. So these L and D forms, why some of them are active? Because this tells you about how that particular group, in which direction, so it tells you directionality of the functional groups or moieties. So if the functional groups are present in such form that there are lesser steric clashes, so that particular form is more favored than the other. Given that, if means, for example, in this case, the NAC is typically present in L form, whereas NAM, it exists in the D form. So if there is certain reason or functional significance behind that particular form, so in such scenarios, that particular group or particular molecule or biomolecule can exist in that form. So in this case, NAM is existing in D form, although L form are more favored. So due to this functionality, due to this cross-linking, we require the D form of NAM. So this specific chirality forms, they are essential for proper assembly and stability of cell wall structures. So as we can see that D-alanine is also helping you to link this particular NAG which contain this tetrapeptide to another NAG containing tetrapeptide. So glycine can only link to this D-alanine. So that's why it is responsible for this interconnections or cross-linking, this particular conformation. Okay, so these amino acids in this peptidoglycan, they exhibit chirality. So as we have seen that there are certain D amino acids as well as certain L amino acids. So these D amino acids such as D-alanine, which we have seen, so which is at the terminal point. So it is non-natural in nature, but it is crucial for the cross-linking. So that's why this is favored in this peptidoglycan. And this D-amino acid incorporation is essential because it also prevents enzymatic degradation. So the enzymes such as peptidases or other enzymes which are present in our body, so they generally terminates, or you can say they degrade the terminal amino acids. So these enzymes, they recognize L form of the amino acid and degrade them. So if the amino acid is present in D form, then these enzymes won't react. So that's why they also avoid the enzymatic degradation. They prevent the enzymatic degradation because of this D form. So that is also one of the reasons that why D form of amino acid is favored in peptidoglycans. And this chirality of molecules means of amino acids and sugars, it also ensures that there is rigidity in the structure. Because of cross-linking, it will be rigid, as well as functionality of this peptidoglycan mesh, so which allows the bacterial cell to maintain its shape. So this cross-linking is also helping it to maintain certain kind of shape, as well as it is also providing resistance to the osmotic pressure which is generated. So and due to this cross linking, it won't gain up that much water, and there will be lesser, means there will be no such osmotic pressure which could be generated inside the bacterial cell wall. Now the concept of why beta lactams are, means they act as antibiotics. So beta lactam, this is the structure of the beta lactam ring, and all the molecules which composed of this beta lactam ring comes under beta lactam. So penicillin is one of them, one of the example where it contains the beta lactam ring. So what this beta lactam ring can do? So this ring is essential for their ability to inhibit the bacterial cell wall synthesis, making them effective in treating the bacterial infections. So these means Means now in the later slide we'll understand that what is the mechanism, how they are interfering with the bacterial cell wall synthesis. But this is the example. So penicillin comes under the category of beta lactams. This ring is present, which is the beta lactam ring, and this is the structure of penicillin. So when you compare or superimpose this structure with D-alanine and D-alanine peptidoglycan terminus, so these are somewhere similar. So if superimpose them, they means nearly look similar. So what can happen is that this particular D-alanine and D-alanine, it was acting as a substrate for the enzyme which is responsible for the cross-linking. So which I have shown in the previous slide. So now what can happen is that penicillin is also exhibiting the similar type of structure. Now what the structure can do is that because enzymes, they attaches to substrate, they have certain conformations. That substrate binds to the enzyme and makes some product. So in this scenario, now enzyme will get confused that whether the enzyme will think that D-alanine, D-alanine and penicillin, they are the same. So penicillin will bind instead of D-alanine, D-alanine. So now that enzyme won't be able to perform cross-linking, so due to which it will interfere with the bacterial cell wall synthesis. So now this is the mechanism of action, that what is happening. So this PBP enzyme, which is I think penicillin binding proteins. So these PBP are the penicillin binding proteins which binds to penicillin. So in the normal scenarios, they will bind to this tetra, or so we can say the pentapeptide which is present. So they will bind to them and they will bring the trans glycosylase subunit which is responsible for the cross-linking. So with the loss of terminal DLA, there will be cross-linking which can take place. So this is the condition where there is no penicillin. This is the normal condition where the cell wall synthesis is happening in the microorganism. But what can happen if penicillin is present? So as we know, this green circles, they represent penicillin. So this penicillin's structure was nearly similar to that of D-alanine, D-alanine. So instead of this PBP binding to that D-alanine, D-alanine, it is now binding to penicillin itself. So it is not going further, due to which the enzyme transglycosylase subunit of that particular enzyme and this PBP, now they will not reach here, due to which there will be no cross-linking between two different sugar moieties and the pentapeptide which was present. So it will break the bonds. So by interfering with this process, now beta-lactam is causing the weakening of cell wall because the strength was provided by the cross-linking. So now no cross-linking means bacterial cell wall will weaken, and which will lead to the lysis of the cell and death. Because we have also read that this peptidoglycan, this particular arrangement of peptidoglycan is also preventing osmotic stress, and means other functionality was also present by this peptidoglycan. So as this cross-linking no longer happens, so it will means bacterial cell wall will lyse, it will break down, and then bacteria means other means environmental molecules will attack that bacteria and it will die. So due to which these beta lactams, they have this antibiotic property due to this particular interaction, because they exhibit similarity with the D-alanine of this particular pentapeptide moiety. So this is the reason that why they are antibiotics. Now we will talk about the levels of protein structures. So is it clear till now? Okay, we'll see next. So now we'll talk about the levels of protein structures. So first is the primary structure. So primary structure means when you are only looking at the sequence of amino acids. Secondary structure means when that particular sequence, when it forms some hydrogen bonding among different amino acids and other groups, then they will take some structure. So that comes under secondary structure when different binding interactions come into picture, such as disulfide bonds or other interactions. So they can take form of alpha helix or beta sheets or loops, turns. So all those comes under secondary structure. Then comes tertiary structure. Tertiary structure means when there is defined 3D shape of that particular protein. So that is the 3D structure. Quaternary structure means when multiple polypeptides are present and they are connected to one another. So what can happen is that a particular protein can have different peptides, different chains. So when those all the chains are combined and a bigger structure is formed, that is quaternary structure. So these are the different levels of protein structures. So now instead of talking about primary structure, which is the normal amino acid sequence, we'll move to the secondary structure where the another interactions come into picture. So these are the local folded structures. So local, why local? Because it is not the global folded structure, it is local. So at local regions, what is the structure? So that's why it is the local folded structure which is formed by the polypeptide chain primarily through hydrogen bonding. So hydrogen bonding is the main interaction which is observed in the secondary structures, due to which these structures achieve the stable three-dimensional shape, and it is also essential for their functions. So now different types of secondary structures. First one is alpha helix. So alpha helix, and in alpha helix also the right-handed spiral is more favored. So it is formed by the hydrogen bonds present between C double bond O of one amino acid and NH of the another amino acid, and which are four residue apart. So in the next slide, we'll see that I and I plus four. So in there, CO means C double bond O, it interacts with the NH group. So oxygen and hydrogen, there is interaction between oxygen and hydrogen of them. So this is forming the hydrogen bond. And then example of the proteins which show this alpha helix are the keratin and myoglobin. Then the next secondary structure is the beta pleated sheet. So there is a difference between sheet and strand. So when there is single strand or single means beta sheets are means beta strands are represented by the arrow-like structure. So when there is a single arrow, then it is a beta strand, and if multiple such strands come into picture, then they form the beta sheet. So it will be like sheet-like structure. So these are the strands of polypeptides which are arranged or which are aligned side to side, which are connected by the hydrogen bonds. They can be parallel depending on the means directionality of them. So they can be parallel or anti-parallel. And the example of the proteins which contain the beta pleated sheets are silk as well as certain antibodies. Then comes turns and loops. So these are the flexible regions that connect different secondary structures. For example, if I want to join two alpha helices, then there will be certain loops or certain turns which are present. So in this figure, the alpha helices are shown in the red color. So these are the spiral right-handed alpha helices. And then these green means arrow-like or ribbon-like structures which are shown in green. So these are the beta strands. And if multiple beta strands they are means next to each other, so they are forming a beta sheet. And then they also have a sense of directionality. So the direction of arrow is telling you that in which direction they are running. So if two alternate strands they are in the similar direction, then that is parallel strand, and if they are in anti-parallel direction, then they are referred to as anti-parallel beta sheets. And these yellow are simply the bound molecules, because proteins also interact with other molecules. So this is just to show them. And then loops and turns in gray. So all these loops which are connecting to different types of secondary structures or same type of secondary structure. So two secondary structures are connected to each other by these loops as well as turns. And the difference between loop and turn is just very little difference, that in turn the lesser number of amino acids are present. If the amino acids which are connecting to secondary structure are large, then they will called as loops. Okay. Yeah. Sorry, what can we understand? Mhm. Okay. The reduction of amine means alpha helices. So reduction, alpha helical content. So reduction, you mean to say that when different amino acids are present, then they are not adopting alpha helical structure. That you mean by the reduction in alpha helix content? Because this alpha helical structure, so this is adopted because of certain molecules which favor this particular conformation. So with loss, you mean to say that if that particular amino acids are lost due to which this alpha helical content is reduced, or you want to say that in certain proteins we see lesser of alpha helices and more of beta sheets? Since you are talking about the same protein which is undergoing reduction of alpha helices, or different proteins which have variation in the alpha helix content. Okay, so these means what I can say is that these alpha helices and beta sheets. So when alpha helices are present in the greater concentration or greater amount, then they leads to the globular structure of the protein. So due to which this alpha helices are also responsible for interaction with different uh, other proteins as well as the DNA molecule also. So there are different interactions in which the alpha helices and beta sheets participate in. So mostly alpha helices are responsible for the globular form of the structure of the protein, and if there are more number of beta sheets, then they will give rise to the fibrous protein structure. So yeah, yeah, you're audible. Okay. So alpha helical interaction with other molecules means these small molecules or other. Uh, what do you mean by other molecules? Okay, so I mean, okay, so what I have got from your question is that you are meant to say that if these molecules are interact performing certain interaction with other molecules, then this alpha helix content is getting reduced. So means so the yeah, I got your point, I guess. So means when we are predicting that what particular secondary structure these amino acids will adopt, so there are certain angles, dihedral angles, which are responsible for the which favors one particular type of secondary structure. So when there are certain interactions, the groups of those molecules, they can also interfere with the fold or the secondary structure which a protein can adopt. So in certain interactions, I guess what happen is that other molecules, when they are interacting with those amino acids or positions which are previously involved in the alpha helical formations, so due to those interactions, now that particular molecule is not in the favorable orientation to take the alpha fold structure. So due to which I guess there is a reduction in the alpha fold content. So that means I want to say that for the formation of particular secondary structure, there has to be certain means favorable angles or certain favorable means structural forces should be present. So if interactions will happen, then they can interfere with the fold of that particular protein. Because for example, in the alpha helical structure, which I'll discuss in the other slide also, that how the R groups and means other groups are present. So if there is interaction, that will bring about certain other groups also. So what can happen is then now there is a possibility that there will be certain steric clashes, which can ultimately, I guess, reduce the means favorability of alpha helical structure, due to which the alpha helical content can reduce. Yeah. Yeah, so I want to say that there might be certain interactions which is interfering with the stability of alpha helical form. That could be steric clashes or other means if there are larger molecules. So it depends that what kind of interaction is interfering. That can lead to the reduction of the alpha helical ability or alpha helical content of the protein. Yeah. And maybe in the later slides it might become evident or clear. So yeah, so this is just the representation that how the beta pleated sheets appear and how the alpha helical sheets are available. So yeah, so in this also I have shown that how the R group and this C double bond O is present. So in different directions, because if they are present, if R group instead of the hydrogen present on the top, if R group is present on this topmost side and this double bond O is present in this side, so these O and R they will means there will be steric clashes among them. So that's why this particular orientation is adopted. And this C double bond O and NH, they are present in the trans conformation. So that means both the groups of this hydrogen and double bond O, they are present in opposite directions due to which to reduce the steric clashes. So maybe if some other molecule will come, they will interfere with these interactions. They will break these interactions. For example, in alpha helical structure, these hydrogen bonds are very responsible. So for example, if I and I plus 4 amino acid, that is making the hydrogen bond. So if some other molecule will come and it will interact with C double bond O. So now this hydrogen bond will break. So now if hydrogen bond will break, then it won't adopt this spiral structure. So this could also be the reason that why the alpha helical content or alpha helix will get reduced, its size will decrease because of the breakage of hydrogen bonds. That is also one of the reasons that if hydrogen bond is broken or if means it is not formed, then it will also lead to the it will interfere with the secondary structure which is formed. Similar case in the beta helices also. And in this also, there are hydrogen bonds which are linking these two beta sheets which are running either parallelly or anti-parallelly to each other. So if there is any breakage in these hydrogen bonds, then the means this beta sheet content will also reduce. Okay, now we'll talk in detail about the alpha helix. So in this case, the journal alpha helix, which is which is more observed. So that is the helix which contains 3.6 residues in one turn. So in this one turn, there will be 3.6 residues, and the distance between each residue, which is also known as the rise, that is 1.5 angstrom. So rise means distance between two residues, and we know that there are 3.6 residues in the turn. So we multiply them, which will give us the distance or the pitch of the helix. That means you can say the height of the helix or height of the turn. So that is 5.4 angstrom. So this particular height is 5.4, which is calculated by multiplying the number of residues to the rise per turn. So I guess there is one question in your assignment also where you have to find the height of the alpha helix, and you are given the number of residues, and you know that the rise is 1.5. So simply you can calculate the height by simple multiplication. Okay, so now the main governing means interactions which will decide that whether means these amino acids or this particular peptide will adopt a alpha helical structure is the bond between this CO group at the CO group at the N position to the NH group at the N plus 4. So there is hydrogen bond formation among these groups, and in the entire alpha helix you will observe this I and I plus 4 hydrogen bonding among all these residues. So this is also one of the property of these alpha helices, and they are generally existing in the right-handed helix form, which is more favorable, structurally stable, and majorly observed. So the amino acids which are good helix formers, they are alanine, glutamine, leucine, and methionine, whereas the amino acids which are less favored in such alpha helical structures are proline, glycine, tyrosine, and serine. Okay, so now these alpha helix can also exist in different forms. So one is alpha helix, which is majorly observed, which we have discussed. Then comes 310 helix and pi helix. So these are the rare forms, but they can be observed but lesser. So at 97% times, you will see the alpha helical structure exist in this form in which the amino acid per turn are 3.6. So there are 3.6 amino acids present in one turn. In 310 helix, you will see there is only 3 amino acids per turn, whereas in pi helices, we have 4.4 amino acids in each turn. Their rise will be same, that is 1.5 will be the distance between two residues. So, and in journal, these alpha helices are about 10 residues on average, and the side chains are well staggered, preventing the steric hindrances. So, as I mentioned, that to avoid those steric hindrances, also these R groups or the side chains, they are means oriented in opposite to that of the C double bond O group. So, to maintain the proper interactions and avoid steric clashes. So, helices can also form bundles or coiled coils. So, there are different types of means when we'll talk about the super secondary structures, then we'll see that in different forms the helices can be present, such as bundles or coiled coils. So now this is about the beta sheets. So as we have seen that in beta sheets, there are multiple beta strands which are running in different direction to each other. So this is the highly extended form of a polypeptide chain. So this one is the extended form. In alpha helices, that was spiral, so that was means not that much extended. But in beta sheets, this is the extended form of polypeptide, and each beta strand can typically have 5 to 10 residues, and distance between two adjacent amino acid is 3.5 angstrom. So in the previous it was 1.5, whereas in this case it's 3.5, due to which it is extended. Then adjacent side chains point in opposite directions. So as in this case, these red dots are representing the R groups. So you can see that whenever the two amino acids are linked to one another, their R groups are in the opposite directions. And these R groups are now involved in forming the hydrogen bonds with the another beta strand. So these are two different beta strands in which the adjacent amino acids have the amino means R groups projecting in different directions to avoid the steric clashes or steric hindrances. So hydrogen bonds and the hydrogen bonds which we observe, they are not at the optimal angle. So there is no such optimal angle which is present. And this is the anti-parallel and parallel beta sheets. So in case of anti-parallel, two beta strands, they are running in opposite directions. So means how this directionality is just, so N to C terminal. So N to C is means directionality of a protein. So when these polypeptides, or you can say when these two different um, peptides, they run in opposite directions, then that forms anti-parallel beta strands. And if they are running in the same direction, that is N to C in both the sides, then it is the parallel beta sheet or parallel beta strands. And the third type of secondary structure after alpha and beta helices are the loops and the turns. And their role is just to connect those two types of secondary structures. So loops are usually, they contain the hydrophilic residues and they are found on the surface of the proteins, and they connect alpha helices and beta sheets. They can even connect two alpha helices as well. Then comes turns. So turns means the loops which have less than 5 amino acids. So then they are known as turns. And if this particular amino acids are large, then they are known as loops. That is only the one of the difference. And then beta turns are common. So there are means the molecules which are combining two different beta subunits, they are known as beta turns, and these are more common. We don't see much of the loops joining two beta strands. They are generally the beta turns. So they are lesser means residues which are joining two different beta strands. They are common and they occur frequently whenever the strand in the sheet changes its direction. So in the as we have seen in the sheets, there are multiple beta strands which are running. So those beta strands, they are connected by these beta turns, and the 180-degree turn is accomplished over the four amino acids. So these four amino acids are sufficient to make that turn, and this is stabilized by the hydrogen bond from the carbonyl oxygen to the amide proton, three residues down the sequence. And the proline in position 2 and glycine in position 3 are common in turns. So in beta turns, the amino acids which are more frequently observed are the proline at the second position and glycine at the third position. So this provides them ability to make that turn. So this is all about the secondary structures, that how different secondary structures are present and how the loops and turns are helping them to connect two different types of secondary structures. Now we will talk about the tertiary structure. So secondary structure was the local, when we are looking at the local polypeptide structure. But when we are looking at the global structure of an entire peptide, then it is the tertiary structure, when multiple other interactions will also come into picture. So in secondary structures, we only focuses on the hydrogen bonds. But now in tertiary structure, it is the overall spatial arrangement of atoms in a protein. So it is stabilized by numerous weak interactions. So when we say weak interactions, which means the non-covalent interactions. For example, hydrogen bond, this is also a weak interaction. Then ionic bonds, which is present between two oppositely charged molecules. For example, in this it is positively charged and this amino acid is negatively charged. So lysine and aspartic acid. So they are forming the interaction which is known as ionic bond. And in certain literature, you will also see that they are referred to as the salt bridges as well. Then there are hydrophobic interactions which are present between two non-polar amino acids such as alanine and valine. And then there are certain hydrophilic interactions, such as serine, which is polar in nature. It has OH group, so it interacts with external aqueous environment, such as water. So these are forming hydrophilic interactions. So all those interactions, they are governing that how this particular protein will fold. What will be the structure of the protein? So this is referred to as tertiary structure, where these all interactions come into picture. And I've not talked about disulfide bond, which is the covalent interaction. So disulfide bonds, I mean, they are not that much frequent in the amino acid as compared to the other interactions. And these are only observed between two cysteine groups, two cysteine amino acids which are present. So these come under the covalent bonds. Along with this disulfide bond, the another covalent bond is the peptide bond which is present. So these are two type of covalent bond interactions which are present. in the proteins. So these weak interaction between the amino acid side chains, they are largely hydrophobic as well as polar interactions, and they can be stabilized by disulfide bonds. So interacting amino acids are not necessarily next to each other. So as you can see that in this case, the hydrogen bond which is formed, it is not formed between two adjacent amino acids. You can see that this particular serine is lying so far from this asparagine. So that means these weak interactions, they can be present among the amino acids which are lying at far away positions. So it is also possible that means the initial or the N terminal amino acids, they're interacting with certain C terminal amino acids. So in that situation, they will bring the N and C terminal of the protein close to each other. So that is one of the reason that how the primary sequence or what amino acid which are present, they can determine that what fold a protein will take depending on these interactions. So two major classes of protein tertiary structure are fibrous and globular. And now we'll talk about the quaternary structure. So this fibrous and globular, it also depends on the what type of interactions are present or what type of secondary structures are present. So in case of globular proteins, you'll see more of the alpha helices which are present. This will be a well-defined structure, and it will be a, you can say, a constricted structure in which all the units are tightly packed. Whereas in case of fibrous proteins, they contain more of the beta sheets, that is more of the extended structure. They will be like a fibers, long fibers, and not means coagulated or means constricted structure. It will be the extended fiber-like structure. So that is fibrous tertiary structure. And when we talk about the quaternary structure, so it means that if multiple polypeptides, for example, hemoglobin, it contains 4 different chains, so that are different polypeptides, that is multiple subunits. So in such scenarios, whenever the structure is formed by interaction of different peptide units or different chains, then that structure is quaternary structure. And this quaternary structure is formed by the assembly of individual polypeptides into larger functional clusters, and this refers to the number of subunits and their relative positions and contacts. So these subunits can also interact with one another with certain contacts, and there could be different forces which are involved in the formation. So such as salt bridges, hydrogen bonds, van der Waals forces, disulfide bonds. So they can also means disulfide bond can also be present between two different uh, peptides as well. So these interactions are common, and then other interactions also come into picture. So in this case, as I have shown you subunit 1 and subunit 2, so these are different types of secondary structures. The combination of secondary structure which are present along with other interactions which are present, so they are forming the tertiary structure. Now two different tertiary structures, that means these are two subunits of a single protein. So now how they're interacting with one another. So these are representing these disulfide bonds. So when we are studying this type of interaction and structure, this is referred to as a quaternary structure. So with each level, we are adding new information to the protein structure. So most of the studies, they provide you the tertiary structure. In certain cases, you'll also find the quaternary structure. Yeah, so now I'll talk about the difference between the fibrous and globular proteins. So this is the globular protein, and this extended one is the fibrous protein. And hemoglobin is a globular protein, whereas collagen, it exists in the form of the fibrous protein. So globular protein have roughly circular shape, whereas fibrous protein, they are like the long strands. The amino acid sequence which is present in the globular proteins, they are irregular and wide range of R groups that could be present in the globular proteins. Whereas in fibrous proteins, they have the repetitive limited number of R groups which are repetitive. And the function is that in globular, it has physiological role or functional role, that is certain enzymes, immunoglobulins, insulin, hemoglobin. So these are certain functions. So the globular proteins are observed in the proteins which are exhibiting certain functions or have certain physiological properties, whereas fibrous proteins, they are generally involved in the structural part of the organism. So when the structure formation is there. So for example, collagen, keratin, myosin, actin, fibrin. So these have the structural role in our body. So in such scenarios, we observe that they have fibrous structure. If we talk about solubility, then these globular structures, they are soluble in water, whereas these fibrous are insoluble in water. So this is the main difference among the fibrous and globular proteins. Yeah, so now we'll move to the another detail, which is the peptide bond formation. Now we will see that how these different structures are actually adopted. What are the forces which are governing that in which conformation the protein will be present. So first of all, we need to understand that how this peptide bond is formed to move on to the concept of folding. So now peptide bond, it is formed between the carboxyl group of one amino acid with the amino group of the another amino acid. So these are two amino acids. And then for the formation of peptide bond, which is also known as CONH bond, there is loss of water molecule. So now water molecule is lost and this peptide bond is formed between this carboxyl group and amino group. So CONH bond. And this is also known as a condensation reaction because there is loss of water. And this bond links amino acids in the protein chain and forms the backbone of the protein. Now this we'll also talk about the partial double bond nature, and we'll also see that why this bond is the bond which do not rotate or there is no such movement across this bond. So what is the reason that this peptide bond, it has partial double bond character. Now how this partial double bond character arises? So because this nitrogen also contains a lone pair of electrons, so in this scenario it shows resonance. So due to resonance it can exist in these, it is reversible, so it can exist in these different forms where this lone pair is transferred to the C. So in this case, there is a partial double bond between C and N group, due to which this C and N will try to come closer to each other. So in the normal amines, this CN bond length is slightly larger than this particular CN bond length in case of amino acids. So due to this partial double bond character, so CN bond length, yeah, so this I already discussed. So oxygen has partial negative charge and nitrogen has partial positive charge, due to which there is an electric dipole. So, and this is also one of the reason that this peptide bond does not rotate due to this uh, partial double bond character. And this bond is also known as the omega bond. So in the next slide, we'll see. Now these peptide bonds, they can exist in cis form also and trans form also. So trans form is more common. So what is the trans form? Where the O and H, they are in the trans positions or they are facing in the opposite directions. They are not next to each other. So this is known as trans form. And why it is favored? Because in this there are no steric clashes or no such interactions. O is getting enough space to occupying space. Whereas if we talk about the cis form of this particular peptide bond. So in this case, the O and H, they are present towards the same direction. Now what is happening with this? If O and H are present in the similar direction, firstly they can also interfere with one another. And now this R group is also, these two R groups are also coming closer to each other. So these side chains, they will also means exhibit the repulsion forces. There will be steric clashes. So due to which this cis form is not favored. There are certain exceptions in which this form can exist for certain amino acids, but normally you will see that this peptide bond is present in the trans form, and this is more stable due to lower steric hindrances. And overall, 90% of the peptide bonds in the proteins, they adopt this trans form. And cis form is found mainly in the proline containing peptides. So 10% of proline containing peptides, they can show the cis form because proline's rigid structure reduces the energy difference between the cis and trans form. So due to which it can take in rare scenarios cis form. So now let's move to the dihedral or torsion angles, which are phi and psi. So after peptide bond, we have discussed that that angle is the omega, and they does not rotate due to the partial double bond character. But the another bonds which are present, for example, among N C alpha and C alpha and the C beta, so they have still the ability to rotate. So when we talk about the torsion angles, these are not just the simple angles between N, C alpha and C beta, but these angles are formed between the intersecting planes. So first of all, we construct a plane. So when two planes are constructed, we look at the site where the intersection of these two planes are happening, and then we draw a normal to that plane, and then we find that in which direction the rotation is happening. So that is how these bonds are calculated. So it describes rotation between two planes along a single bond in the polypeptide backbone. And these torsion angles are also known as dihedral angles, and they are simply represented by the symbol tau, which measures orientation of four sequentially linked atoms in a protein backbone. So whenever we will write, we'll not simply write C alpha, C beta. We will write as N C alpha C beta. And now you can see that this bond is not between these three. Although we are writing N C alpha C beta, but it is not between these three uh, atoms, but it is between the plane. So, for example, in this case, N C alpha and C beta. So this particular plane is means this entire plane when it's forming bond between N C alpha and C. So this is known as phi, where the bond is formed between nitrogen and C alpha. Whereas if this bond is formed between C alpha, C beta and another nitrogen, then this C alpha and C beta, this particular bond is known as psi bond. So to remember it, you can remember it by, for example, CC. So psi. C and N is phi. And then many people confuse this C alpha and N bond with a peptide bond. So the peptide bond is formed between N and C beta because it is formed by the carboxyl carbon and amino group, not the C alpha carbon. So in this case, this angle is the omega bond. So if we'll find the bond between this plane, it will be omega. And the bond between N and C alpha, that is phi. So this is the difference between phi bond and omega bond. And omega bond is the peptide bond is fixed, it will not move, it is rigid. So due to which we do not put much attention into it. Okay, so now the angle between the planes formed by N C alpha and C prime is also the C beta only. So C prime means the carboxyl carbon. And then the plane formed by the C alpha, C prime, that means C beta and N. So this is the another, which is psi angle. And then along with this omega is also present. So these different angles, they determine the protein folding and the secondary structure because these can rotate. So they will determine that in which position. So because their rotation can, so in the next slide, I'll show you that how their rotation can influence the orientation of these R chains, R groups which are present. So this orientation, they can move in certain angles, but if one particular angle in which the R groups will become closer to each other, then there is a possibility that they will interact or repel each other. So that will be disfavored or disallowed angle. So due to which the scientist named Ramachandran, he has given the angles which are favorable, where there are least clashes and more favorable interactions are present. So now this is the reason that why we are studying these phi and psi angles. Okay, so as I've told you that phi angle is between N and C alpha, and psi angle is between C alpha and C beta, which is C prime, and omega angle is between C beta and N, which is peptide bond, and it is usually fixed at 180 degrees, that is trans. So it is between the plane CO and H. So these CO and H, that plane, the angle is 180 degrees, which is the trans form. Due to its partial bond character, it is fixed. And cis, that means in the case where the angle is 0 degrees, which is mostly in Prolian. Okay, so now here I'm showing you that how the phi and psi angle are varying. So this is how the rotation happens. We'll move entire plane. So now you can see that if these particular R groups are moving in certain directions, so it can rotate in different directions until and unless there are no steady clashes. So those such angles will be known as the allowed regions, and it depends that which one plane is moving. For example, if C alpha and nitrogen plane is moving, then it is known as the phi angle, and the if C alpha and another carbon is moving, then it is known as the psi angle. So Ramachandran plot. So G.N. Ramachandran was the one who performed dedicated research in this, and he came up with a graph which tells you the angles which are the allowed and disallowed torsion angles of amino acids in the protein, and these are very helpful to know that in which what will be the particular fold of the protein, how it will get folded, what will be the secondary structure, because those amino acids which are present, they determine that whether it will adopt the alpha helical structure or whether it will form the beta sheet. So all those things are very important, and these psi and phi angles will tell you that in which form particular amino acid will exist, or particular protein, what form it will take. So now in this graph in Ramachandran plot, it tells you about the allowed regions. So for alpha helices, they are found in the specific region. For example, this particular region where the phi values are and psi values are positive. So these are rare because as we discussed that alpha helices mostly exist in the right-handed form, but they exist in left-handed form when the psi and phi angles are positive. This is the landscape or the position where these are the angles where the peptide can exist in the left-handed alpha helical form. This is the allowed region. If we talk about right-handed alpha helix, then these are the means it will exist when the psi and phi angle both are negative, when the values are negative to each other. So these are found between the range minus 60 degrees. Means phi will be minus 60 degree and psi will be minus 50 degree. So these are the regions where the right-handed alpha helix are more allowed. These are the favored angles. Then if we talk about beta sheets, then these particular psi and phi values are the places where the beta sheets are allowed. These are the favorable angles. So they range between the phi is nearly minus 120 degrees and psi is plus 120 degrees. So in beta sheets, the psi values are positive and phi values are negative. Whereas left-handed alpha helices, they although appear in the rare regions with the positive psi and phi values. Then comes disallowed regions. So all other regions which are not highlighted with any color, they are disallowed regions. And why they are disallowed? Because they will lead to certain steric clashes. Because the movement of the plane is also moving the R groups and other groups in the present in the amino acid. So those R groups, when they are present in closer to one another, so there will be steric hindrances. That's why those angles or those regions, they are disallowed. Then proline and glycine, they show certain exceptions. For example, proline, it has restricted phi due to its rigid ring structure. So there is some restricted phi angle only which this particular proline can take due to its rigid ring structure. And if we talk about glycine, as we know it is achiral, it contains two hydrogens. It in R group there is no another bulky group which is present. So it is more flexible because if it is present then there are means least chances that its group will interfere with another group. So that's why it can occupy a wide range of psi and phi angles. So this is also one of the reasons that why glycine can occupy a different region in the Ramachandran plot. So now this is the same thing is represented in the particular type of beta helices and alpha helices. So beta sheets and alpha helices. So these are angles. For example, this particular psi and phi values, they are responsible in case of antiparallel beta sheets. So these are the psi phi values where the antiparallel beta sheet are more favored. Whereas if this particular psi and phi value are present, then parallel beta sheets are favored. So in this way, you can see that these psi and phi value calculations are telling you that what is the favorable secondary structure. And these values are very helpful because as we know that in currently the different tools have come up which predict the structure of the protein. So in the primitive time, when this alpha fold was not present, so they used to look for the Ramachandran plot and look for the angles favored or means allowed and disallowed angles, and then predict that what would be the secondary structure that particular peptide can take. Even now, also Ramachandran plot validation is very important to check that whether the structure which is predicted, whether it is lying in line with the Ramachandran plot, whether it satisfies the criteria of Ramachandran plot or not. So this is how the psi and phi values determine that what structure a particular protein or particular peptide sequence can take. Now let's talk about the super secondary structure. So on above secondary structure, we have super secondary structure, which is the combination of secondary structures arranged in specific geometric patterns. And these are the functional building blocks. So key feature of super secondary structure is that they are building blocks of protein folding. They have common motifs. So when multiple secondary structures are combining, they give rise to motif. Motif means when there is a recurring secondary structure in particular pattern. That is motif. And domain means if there are larger functional part which is independent. So that is domain, which is the larger motif. If multiple motifs are present, they might form a domain. So common motifs are four helix bundle, beta meander, Greek key, rose manifold. So these are the different types of motifs. So in motifs, what you can see is that there are multiple secondary structural elements are present, and the structural role is that the super secondary structures they bridges the secondary and tertiary structure because they are providing the folding to particular protein. So in this case, there are multiple helices which are running anti-parallel direction to each other. And then there are another four helix bundle with two anti-parallel and two parallel helices. So these two are parallel and these two are parallel to each other but anti-parallel to these two. So in this way, these are different types of motifs which are commonly observed. So what is the difference among motif and domain? So motif means stable arrangement of multiple secondary structural elements, and these will be repetitive. And there will be certain pattern which will be constant throughout the entire protein, whereas domains are the independent and functional three-dimensional self-stable units. So they can also be entire protein or the part of the protein which is functional in itself. These motifs are quite small. They are not functional in itself, but domains, they are functional in themselves. They have proper well-defined three-dimensional structure, and they can also represent the whole protein as well as some part of the protein. Yeah, so these are the representations. So in this case, these domains, they have multiple motifs which are present. Similarly, this also has different means multiple beta, alpha, beta motifs and forming a domain which is functional. And then comes larger proteins which can incorporate multiple domains as well. So proteins can have more than one domain and domains can have more than one motif, whereas motif is the smallest unit. Now, the rules which govern the secondary structures are hydrophobic core formation, layered organizations, stacking of polypeptide segments, and beta sheet stability. So, this is all about the rules which govern that what secondary structure the protein will take. Now we will talk about the interactions. So we have already discussed different covalent and non-covalent interactions. And once again, we'll just quickly revise them. So one is hydrogen bond, which is formed between the polar amino acids. Then comes hydrophobic interactions between non-polar amino acids because they have the alkyl groups, so they shows hydrophobic interactions. Then come disulfide bridges, which is type of covalent interaction. Then ionic bonds or salt bridges between two oppositely charged amino acids. This is the overall three-dimensional structure which is adopted by these interactions. Okay, now we'll talk about the thermodynamics of protein folding. So now how to determine, because if everything is so simple, then why we are struggling with predicting the fold of the protein. So now protein folding means when this polypeptide, it is folding into a characteristic and functional three-dimensional structure from the random structure that is known as protein folding. So normally the protein when it is synthesized, for example, the translation when it happens in the cytoplasm where mRNA is forming the polypeptide chain, then initially it is a random structure. Later on, with time, it folds into a proper well-defined structure. And if that fold is not proper, for example, if it is misfolded, then there are certain enzymes which promote the folding of the protein. So now what are the factors? What is the thermodynamics behind this protein folding? So firstly, if we talk about the stability of the protein or folding of the protein, then the concept of free energy comes into picture. So free energy means that if protein folding is spontaneous, then this free energy, that is delta G, is negative, which is resulting from the balance of two factors. So what are the factors which is helpful in calculating the free energy? So they are delta H, that means change in enthalpy, and delta S, that is change in entropy. So delta G is equals to, this is the bigger formula, but simply it is equals to delta H minus T dot delta S. So delta S is the change in entropy, whereas delta H is the enthalpy, that how much energy is lost or gained. So delta H folding stabilizes the protein through intramolecular interactions, which reduces the enthalpy. So if there are intramolecular interactions, then enthalpy will be less. And what we want is that delta G should be negative. So if delta G is negative, that means the protein folding is spontaneous and molecule is stable. So we want negative value of delta G. And how it will become negative? If we have negative delta H or lesser delta H and higher delta S. So if entropy is high and enthalpy is low, then in such scenarios, we will see that there will be negative delta G, and that means stable folding of a protein. So this is about the how the thermodynamics is playing the role. So now internal interactions, they are decreasing delta H. So they are moving to folded landscape. So this is folded versus unfolded protein. And if T delta S, this is also reducing. So due to hydrophobic effect, delta S is increasing. Hence it will reduce the delta G value. So this is also favoring the folding of the protein. Whereas the conformational entropy. So conformational entropy means when this particular amino acid are joining to one another by taking certain conformations. So when the loss of water molecule happens to form the hydrogen bond, then what happens is that due to loss of that, to perform that reaction, to form that particular angle, there is some energy which is utilized. So yeah, so there is some energy which is utilized also, and this is known as the conformational entropy. So in such scenarios, the value of conformational entropy is less, so which means which is disfavoring the folded state, or which is means which is moving the delta G value towards the positive side. So to means on it to deal with that effect, there is hydrophobic effect, which is increasing the delta S and moving it towards the negative side. So when we calculate the net value of delta G, then it is negative. So that means this minus T delta S by hydrophobic effect, it is uh, overplaying over the conformational entropy. Okay, so this is all about the free energy concept, that how enthalpy and entropy are determining that what will be the delta G values. But before it, there is certain concepts which are present. So first of all, Finson was a scientist in 1972. He told that the amino acid sequence, it determines that what will be the three-dimensional structure of the protein. And he performed certain experiment, which is known as ribonuclease refolding experiment, in which he has shown that how if particular interactions among those amino acids are disturbed, then the folding can't happen. So he has used certain chemicals. So in the next slide, I'll show you that what was this experiment. So with which he has mentioned that sequence alone is sufficient to determine the conformation of the protein. And he also received the Nobel Prize in 1972 in chemistry to tell this. Then comes Leventhal paradox. So this paradox says that there could be multiple forms in which protein can fold, but still protein folding, it proceeds through certain kinetic pathways and it avoids exploration of all the possible states, because there could be multiple states in which the protein can fold. Then how it determines that particular structure it will fold into, and that will be the most stable. So according to this paradox, he mentioned that there is some kind of kinetic pathway, because if it will explore all such folding states, then that will take a lot of time. Then comes the kinetic theory of protein folding, which says that it searches for the most accessible stable state in reasonable time. So that at particular time, the state which is accessible and which is stable, so that is being searched, and not entire all the states are not explored at during the folding of a particular protein. And then folding, there are multiple pathways which are means studied for the protein folding, which are mentioned to be responsible, due to which protein folding happens. So in the next slides we'll see that what are the different pathways which are responsible for folding. So this means rapid formation of secondary structures which will aggregate to form domains, because secondary structures are formed and later on tertiary structures, and so ultimately it begins from the formation of secondary structures, and these are known as the intermediates or molten globules. So one is the final state, and in the middle there is the molten globule state, after which the protein will further fold and get into the entire means least energy state or most stable state. So this is the Finson's experiment, in which what he has taken ribonuclease, and it normally folds into the functional native structure. Whereas if we add 8 molar of urea with beta mercaptoethanol, so this urea, it interferes with the hydrogen bonding interactions, and beta mercaptoethanol will disturb the disulfide linkages. So if those interactions are disturbed, then the protein will be denatured and it will not take particular fold or particular structure, it will not form, because there is loss of disulfide bonds. Because as we have talked that two different amino acids lying far away from each other, they can also come closer and form disulfide bonds or other interactions. So now in this state, because hydrogen bonding and disulfide bonds are they are disturbed, they could not no longer be formed. So this leads to wrongly fold means this leads to the misfolded or denatured RNAs. Later on, they have removed this urea and then they have checked. So they observed that there is wrongly folded or misfolded structure was present. So they added a small amount of beta mercaptoethanol, and then they found that it again get refolded into the native structure. So they are saying that these interactions between the linear amino acid chain or linear amino acids, they are means determining that what will be the fold of the protein, what will be the structure of the protein. So due to which they are means he got Nobel Prize to mention that how this linear sequence is determining the structure of or the tertiary structure of the protein. Yeah, so I was telling you about the different models of protein folding. So there are three main models which have been given. So first is the hydrophobic collapse, which says that there are certain hydrophobic interactions because in the protein there are certain amino acids which are hydrophobic in nature. So as we know that in the cytoplasm there is aqueous environment, and when protein is formed, there are certain amino acids which are hydrophobic in nature. So there will be hydrophobic interactions due to which they will move to the core of the protein and they will try to hide from the aqueous environment. So due to which they will collapse. So hydrophobic interaction will lead to the collapsing of the protein into the compact or intermediate structure. So this is how the rearrangement is happening and protein is getting folded. So this is one model which has been given, that how the protein folds. Now second model is the framework model, in which they have mentioned that firstly secondary structures are adopted. So secondary structures are formed and then these secondary structures are further give rise to motifs and domains, and ultimately the tertiary structure is adopted. So this is known as diffusion collision model or the secondary structure formation model or the framework model. Now third model is the nucleation models, which says that these domains they fold independently with smaller subdomains known as structural kernels, which helps the protein to achieve the final structure. So they are mentioning that there are already certain domains, and with the help of subdomains the protein is getting folded. And these are other nucleation with secondary and tertiary structures. So with the condensation, the protein is getting folded. So these are different models which say that how is protein getting folded in the cell. And this is to show you how the energy landscape is changing. So when the protein is unfolded, then its energy is high. That means delta G value is positive. But as we move down, that means if entropy is increasing, the entropy means the randomness among the molecules. So when randomness is increasing, so in that scenarios, the energy values are becoming negative. So this is the molten globule state, which is native state has the least energy. That means delta G value is highly negative in the native state, and molten globule state is where it is near to native state, but means this is not the most stable state, but less stable, not extremely unstable, but less stable state. And after molten globule state, it will enter into the native state. And in this framework also, framework model, they have said that these secondary structures, these when the entire secondary structures are formed for the protein, they are that particular protein at that point of time, it is in the molten globule state, and which get further folded. Certain other interactions also happen, and it finally enters into the native state. Okay, so the key concepts in protein folding, so in order to summarize all of them is, first of all, the hydrophobic effect. Second is the secondary structure formation, which is taking place. Then there are certain interactions which are responsible for the particular folding, such as hydrophobic effect, van der Waals, or certain steric interactions. They also determine that how protein will fold depending on the side chains which are present. So it will determine that what interactions will take place. So on the basis of that, particular folding will happen. And specific interactions such as hydrogen bonds, salt bridges, they are also responsible to determine the fold of the protein. And above all of them, there are certain enzymes which speed up this folding process, such as protein disulfide isomerase, peptidyl proline isomerase, molecular chaperones. So these are certain enzymes which are also allowing the folding of the protein. And now comes the techniques. Now we have seen that this is how the protein get folded, structure is adopted. Now the thing comes that how we can study them, whether how we can quantify them and further perform studies with the proteins which are present. So for quantification, if our aim is to quantify the protein in the cell, means how much concentration of protein is present. So the different methods are Biuret method, Lowry's method, and biosynthonic acid assay, BCA method, and Bradford assay. So there are different chemicals which are present. By using those chemicals, there are different principles that if there is change in the color intensity because these chemicals, they will interact with the proteins and other peptide bonds, and with which they will undergo some chemical reaction, due to which they can undergo change in the color or other things. So with which we can determine the concentration of proteins. So these are different methods in which different reagents are used. Based on them, they have different names. And there is the range, that how much range of protein you can determine with these methods. So these are the methods. and I hope these have been discussed in detail in the class, so I won't go into their detail. Then comes other methods such as gel electrophoresis or isoelectric focusing, which separate the proteins based on their isoelectric point. So with this, what is the advantage of performing isoelectric focusing in that? For example, if you obtain certain protein and you want to know the properties of that protein, that what kind of amino acids are present, so all those properties, they can be studied based on the isoelectric focusing, because we know different amino acids have different pi values. So they will separate the proteins on the basis of pi values. So you will be able to know that what will be the property of amino acid, whether it is composed of mainly the negatively charged amino acids, or positively charged amino acids, or neutral amino acids. So means this is the advantage to understand the content of the protein. Then comes two-dimensional gel electrophoresis. So when we say two-dimensional, that means it is separating those proteins on the basis of two different criteria. So first dimension is the charge. So in this, the separation is happening on the basis of charge of amino acids in the protein. And second is by the size. So as we know, different amino acids have different sizes. So in that way, the separation will happen on the basis of size. So in this also, we can compare the proteins and understand what would be the composition of the protein. Then comes centrifugation. So in centrifugation, we can separate the particles by size. Then zonal ultracentrifugation, which uses a gradient to separate the proteins on the basis of the size or shape. Then equilibrium density gradient, which separates by particle density. And then the methods of protein sequencing, which are the polypeptide cleavage. So cyanogen bromide, it will cleave at methionine, and Edmond degradation. So this we have discussed in previous class also, that how the Edmond degradation will help you to know the amino acids which are present towards the N terminal of the protein. So from the N terminal, we can perform the sequencing of the protein by using Edmond degradation, in which certain chemicals such as PITC is used, and each time it will take one single amino acid from the N terminal with it, and then we will perform the other techniques such as chromatography and identify the peaks and check that what is the amino acid present at the that particular position. So these are the methods where we can sequence the amino acid. And another such method is mass spectrometry. So in this also, we take the protein, break down it into peptides, and then those peptides are passed through the mass spectrometer, where those peptides are further broken into the ions. Those ions are, their mass to charge ratios and intensities are plotted and identified. So this is also for the identification of the types, or you can say the sequence of the proteins. So sequence of amino acid present in the protein, we use mass spectrometry. And in addition to that, mass spectrometry, as it is telling you the mass to charge ratio, it is forming the ions. So it can also tell you the information about the other post-translation modifications. For example, if there is phosphorylation, so there will be some change in the mass to charge ratio. So with that, we can predict that whether the protein which I am studying, whether it has undergone some post-translation modification or not. So there is already some reference mass to charge ratio information which is with us. So by comparing different mass to charge ratios, by comparing different spectrum which is obtained, we can understand that. So mass spectrometry, it helps in peptide mass fingerprinting and the sequencing of the protein. Then locating disulfide bonds. So in the protein, we can even locate the disulfide bonds by using the reducing agents like BTT for sequencing. Then comes proteomic analysis. So for the proteome level analysis, we can perform electrophoresis, followed by MSMS, and then search them in the database. Similarly, phosphorylation site mapping can also be performed using mass spectrometry. So these are the techniques that how we can study the proteins. We can compare among different proteins. We can also understand the quantity of the protein present, and we can also look for the sequence of the amino acids present in the protein. Now we'll practice few MCQs. So is it clear till now? Okay, so now we'll quickly go through the MCQs. So now the first question is that which of the following amino acids have indole ring? So option A is alanine, option B is arginine, option C is tyrosine, and option D is tryptophan. So, okay. So yeah, the amino acid which contains indole ring is tryptophan, and this is the indole ring which is present. Okay, now let's move to second question. So which is, which of the following amino acids have a sulfur atom? So option A cysteine, option B glycine, option C serine, and option D alanine. So this is also quite simple. Yeah, okay. Yeah, so the amino acid which contains sulfur is cysteine. And these are the other amino acids which are mentioned. So cysteine contains sulfur, and if methionine was present, then methionine also contains sulfur. So next question is, which of the following have a remarkable similarity with penicillin? Option A D-glutamate D-tyrosine, option B D-valine D-alanine, option C D-alanine D-alanine, and option D L-glutamate and L-tyrosine. This also we have discussed. Anybody wants to answer? Okay, let's see. So this one is D-alanine D-alanine, and we have seen that how the penicillin is acting as a antibiotic because it is interfering with the D-alanine D-alanine which is present in the peptidoglycan terminus. So this is how they have remarkable similarity, due to which the penicillin binding protein, which initially binds to D-alanine D-alanine and performs the cross linking, will now bind to penicillin. So cross linking will be affected. Now next question is, which of the following destabilize the alpha helix? A serine, B glycine, C proline, and both B and C. So you have to tell that which of the following amino acid destabilize the alpha helix. Okay, so uh, actually both proline and glycine both are responsible to destabilize the alpha helix. This is the structure of glycine, in which it has hydrogen in the R group. So due to which they are means it can adopt and it can means rotate in any conformation. So when glycine is present, it will also break or means destabilize the alpha helix. And when proline is present, so this also has the bigger ring, and then NH group is present, which is also responding, which will also disturb the alpha helical structure because it will interfere with certain hydrogen bonds which need to be formed. So these both destabilize the alpha helix. And in this, I have given a short mind map, which will help you to understand the concept in better way. So in this, I have shown that how the alpha helix is formed, what are the different types of secondary structures, and this is the right-handed alpha helix. And these concept we have already discussed, that there is 3.6 residues per turn, and each amino acid rotates the helix at 100 degrees. Typical length is 10 amino acids. So at least there should be 10 amino acid in the peptide to call it a alpha helix. And then hydrogen bonding, which is present between the hydrogen bond donor, that is the NH group, and hydrogen bond acceptor, that is the CO group. And in this, hydrogen bonding is formed between the amino acid I plus with the I plus 4 amino acid. And these are the 5 common amino acids which are observed in the alpha helices: methionine, alanine, arginine, lysine, and leucine. Whereas unfavorable amino acids are proline and glycine. So next we'll see that why proline and glycine are unfavorable. Other than this, serine, aspartate, and asparagine, they are also unfavorable. Whereas threonine, valine, and isoleucine are also unfavorable in alpha helices because of the size, charge, and shape of the side chains. So these are also the unfavorable amino acids. Now, why proline is a helix breaker? Because it has the amino group, which does not have hydrogen atom to be donated. So now, if it do not have hydrogen bond, means hydrogen atom, so this will not allow the alpha helical structure to form the proper hydrogen bonding. So due to which it will break the helix. But we can also say that proline, due to its that ring-like structure, it can be present at the terminal of the alpha helix. So if it is present on the terminal position, then it will provide the means rigidity to the alpha helix. So it is good to have proline at the terminal position and not in the middle, because when it is present, it will break or yeah, it will break the alpha helix or destabilize the alpha helix. Whereas if we talk about glycine, then it can take many angles of rotation. It can rotate in any angle because there are no such steady clashes. So due to that particular movement, amount of variability which it brings, so it will be energetically expensive for the glycine to adopt the alpha helix structure. So due to which these both comes under destabilizing the alpha helix. And then these are the example of beta branched amino acids as well as polar amino acids. Yeah, so this is just a mind map to, these are just all the concepts at one place about the secondary structure. Now moving on to next question. So how many amino acids are present per turn are found in the pi helix? Option A 4.4, option B 3, option C 2.8, and option D is 3.6. So pi helix is the rare helix. So any one of you remember? Okay, now let's see. So there are 4.4 amino acids which are present in one turn in pi helix. So this is the pi helix, and in this is also the representation of pi helix. So you can see that how the alpha helical structure is present, and whenever the pi helix comes, this you can see there is a little bit of opening up of this alpha helical structure because there is one additional amino acid which is present. So this pi helix, it is a protein secondary structure which was discovered by Barbara Lowe in 1952, and once it was considered rare, but short pi helices are found in 15% of the known proteins. So means this Barbara, she has shown that this, although this particular pi helices are rare, but still in 15% of known proteins, the smaller portion is observed to be the pi helix. And how this pi helices arise? This arises from the single amino acid insertion in an alpha helix. So if single amino acid comes, then it lead to the formation of the pi helix. And though it is destabilizing, but they are often retained near the functional sites, which means that if they are retained, then they might have some advantage. So that's why they are favored throughout evolution. So this is one of the their speculation that why these pi helices are observed. Okay, so now this is the next question. So you have to choose the correct amino acid which is shown in the image. So whether it is isoleucine, threonine, leucine, or valine. Okay, so this amino acid is isoleucine. So when we say leucine and isoleucine, many people get confused because they have exact same chemical composition and they only vary that how this CH3 means how these methyl groups are arranged. So in case of isoleucine, we see this methyl group is present at the beta carbon. So the carbon next to C alpha carbon is the beta carbon. So this beta carbon contains methyl group and the other ethyl group. So that is the difference between leucine and isoleucine. Whereas in leucine, instead of having this branching at the beta carbon, it contains single methyl group and there is terminal branching. So branching is not happening at this position, it is happening at the terminal position. This is the only difference, that how leucine and isoleucine vary. Whereas in valine, we only have, means in addition to this beta carbon, we have two additional methyl groups, and this is structure of threonine. So in this, we have one OH group with respect to the CH3 present in the valine. Okay, so this structure was of isoleucine. Now, next question is you have to identify the correct dihedral angle of the bond with the red arrow. So you have to identify that what is this bond, whether it is psi, phi, omega, or none of these. But it is between the C alpha and C. Yeah. Okay. Yeah. So this is the psi bond. And this omega bond, I've already told you that it is present between the peptide bond. So when we observe the plane which is forming the peptide bond, then that is omega, which will be between the C beta and N. So C beta is the one which contains COO. This one is the omega, C double bond O with NH. This is the omega, and this C alpha and C is the psi. So correct answer is psi. So now next question is the side chain of two amino acids is around 4 angstrom apart and they have formed a salt bridge. So you have to choose a correct pair, that which amino acids can form the salt bridge. So option A is aspartate and arginine, option B is glycine and alanine, option C is arginine and alanine, And option D is cysteine and cysteine. So this is quite straightforward. Okay. Anybody wants to answer? Okay, let's see. So the right answer is aspartate and arginine. So as I have previously mentioned that these salt bridge means the ionic bond. So it will only be formed between two oppositely charged amino acids. So in this scenario, aspartate contains the negative charge and arginine contains the positive charge. So this negative and positives, these ionic charges, they will form the salt bridge. So this will be the right option. And this is also the another example. So arginine and glutamate can also form the salt bridge. Okay. So this is how we identify that which of the amino acids will form salt bridge. So we only have to find that those two amino acids should have charge and those two should be oppositely charged. Whereas glycine, alanine, they do not have charge. Arginine have charge, but alanine don't have charge. Whereas cysteine, cysteine, none of them has any charge. So that's why option A was correct. Now the second last question. So energies of weak forces, they range from option A is 100 to 200 kilojoules per mole, option B 330 to 100 kilojoule per mole, option C is 200 to 500 kilojoule per mole, and option D is zero point 0.4 to 30 kilojoule per mole. So energy of weak forces. So weak forces here means the interactions, non-covalent interactions. Okay, now let's see. So you might not have any idea. So the right answer is zero point 0.4 to 30 kilojoule per mole. So when we say weak interactions, it involves hydrogen bonds, van der Waals interactions, ionic bonds, and hydrophobic interactions. And these typically range from zero point 0.4 to 30 kilojoule, and these are the individual energy range or energy values. So for van der Waals, it is between zero point 0.4 to 4. For hydrogen bond, it is 4 to 30. For ionic interactions, it is between 20 to 30 kilojoule per mole, and for hydrophobic interactions, it varies, but it is generally weak. So they will lie in the range zero point 0.4 to 30. Whereas you can see the other are quite high. So that's why this is the right option. Now the last question is the formation of tertiary structure of protein is enthalpically. So you have to tell that whether it is favorable, unfavorable, independent or none of these by looking at the enthalpy values. Okay, okay, now let's see. So this is unfavorable. And why it is unfavorable? Because we are only talking about the enthalpy. So enthalpy means delta H. So if it is unfavorable, that means it should have the enthalpy value higher than 0. Because if enthalpy value is greater than 0, then in that scenario, delta G will have the tendency to become positive, which is unfavorable. So why it is enthalpically unfavorable? Because when protein folds, then there are non-covalent interactions such as hydrogen bond, van der Waals forces, and ionic interactions which are formed. But breaking existing bonds with water molecules, or you can say solvation shell, it requires energy. So that energy, when it is energy, it is requiring energy. So that condition, the formation of such hydrophobic cores, disrupt the hydrogen bonds between the water molecules, which leads to increase in enthalpy. So that is responsible to increase the enthalpy. That's why enthalpically these tertiary structures are unfavorable because there is increase in enthalpy. But on the other hand, if we talk about as the net delta G value, so in that scenario, why folding happens? Because other entropy, we look at the hydrophobic effect, so it leads to increase in the entropy. And when hydrophobic residues cluster together, water molecules gain entropy, making the overall Gibbs free energy negative. So which leads to spontaneous folding. So this entropy gained is higher then this enthalpy increase, due to which overall delta G remains negative. So if we talk about the tertiary structure formation, so enthalpically it is unfavorable, but overall it is thermodynamically favorable due to the dominant role of entropy. So that's why this is the answer. Yeah, so this was all about the MCQs. Now I have kept certain add-on concepts. just for your idea and concept building. So just two, three add-on concepts are present. So I'll quickly go through them. So first of all, this is just to show you that with time, how the unfolded state is moving to the protein folding. So this is the time scale, that with time it is folding, and then there are chances that it can also misfold with greater number of time. So in the Okay, so when we see the means molten globule state and the state where it is the native state, which is of the least energy, so negative delta G value. So what can happen is that it doesn't mean that if you take long time for protein folding, then it will get into the native state. This is just to show you that there is a certain reasonable time where the protein adopts the structure which is equivalent to native state. And if the time is increased, if we take more time, then it is a possibility that that protein can get misfolded, and that particular structure is not, stable. So this is the timeline that how the discoveries in protein folding happen, starting with the discovery of protein by Gerardus Mulder, and then Jonas Jacob Berzelius. He named those proteins. Then Linus Pauling came, Robert Corey and Hermann Branson. They proposed alpha helices and beta sheath structure. Then comes myoglobin, which was the first X-ray crystal structure. So for the myoglobin, that first X-ray crystal structure was given. Then Anfinsen came, who demonstrated that how the linear sequence will determine the folding of the protein or the structure of the proteins in 1961, for which he got Nobel Prize in 1972. Then in 1969, Leventhal paradox came up, where they mentioned that how long time is needed to decide a protein to which fold it can take, but whatever is happening, that is taking very lesser time in our biological systems. So in this, they have mentioned that there might be certain uh, kinetic forces which might be acting. Then in 1971, PDB came, and then certain powerful programs came to which predicts the chemical processes in 1977. In 1978, insulin was the first recombinantly expressed protein which was utilized in pharmaceutical industry. Then in 1980s, Sir Greg Winter and Ellen First, they have established protein engineering. Then in 1985, solution NMR structure of the first protein, proteinase inhibitor, was given. So in this case, NMR also came into picture. And then in 1990, cryo-electron microscope structure of bacteriorhodopsin was given. And then in 1995, energy landscape perspective of protein folding came up, that there is a certain molten globule, native-like state. So in this, they have given the energy landscape, that what is the energy in which the protein adopts the stable state. And then in 2021, AlphaFold was launched, which has the ability to predict the tertiary structure of these protein sequences. And now it is still undergoing certain advancements. Now it is also even able to predict the polypeptide structure, that is the quaternary structure in which multiple other uh, subunits of the protein are also coming. Okay, so now as in this entire week, we have talked about primary, secondary, and tertiary structures as well as quaternary structures. So there is another structural, means 5-degree structure, which is also introduced recently, which is quinary. So what quinary means, as in quaternary, we have talked that a single protein can have multiple subunits, multiple chains. So in quinary, what can happen is if two different proteins are interacting, so those two subunits of different proteins can interact and they can also form certain complex. So that is also having some structure. So that particular interactions, when we are looking at structure which is adopted between two different protein subunits, then that is known as quinary, in which there will be different protein-protein interactions. And this can also have certain different types of interactions which we have seen in other structures of proteins. So this is just to tell you that there is a quinary structure which is also introduced. And this is the landscape, energy landscape, that the unfolded proteins, they have higher energy. And as we move down to this landscape, we reaches the native state. And in the middle also, so there is a term known as local minima and global minima. So local minima means that with time, when you are allowing your molecule to get folded, then you might see that if you observe this locally, then you will see that it appears as the local minima. Means it appears minimum for all the time scales for which you are performing the folding, it appears that this is the minimum energy state, and you will think that this is the most stable state. But what can happen is that there might exist some global minima. Global minima means beyond that, there is no such reduce in free energy can happen. That is the maximum stable state present for a protein. So that is known as global minima. So this is the means concept of different funnels or energy landscape model, which tells you that what is the native state or most favorable state for a particular protein to take. And then this particular left side structure represents the folding landscape, and the right side represents the aggregation landscape. So when different protein units are aggregating, then also there is certain decrease in the free energy and they become stable, such as in amyloid fibrils. This is the delta G values. So most negative. Then for oligomers also, and for irreversible amorphous aggregates also. So this is just to show you that how different local as well as global minima exist. And these are different states in which protein can exist. So these are folding intermediates, and this is the native state. Okay, so this was all about today's class. And if you have any doubts, then you can ask about the assignment or maybe anything about the content. Hello? Yeah, yeah, you are audible. Okay. Yeah, I have not mentioned those disordered regions, but yeah, they could be present. So whenever a particular, when we cannot predict the exact structure or secondary structure, we cannot assign to particular peptide, then that comes into the disordered region where the structure could not be predicted. So there are certain amino acids, as I've discussed, that they cannot allow the folding of particular peptide to alpha helical form or beta sheet form. So such amino acids such as glycines or prolines or certain amino acids, so they are mostly abundant in such intrinsically disordered regions, which do not allow the protein to take a stable secondary structure or tertiary structure. So you have any specific doubt about disordered region? So maybe in the next slide I will add that disordered region also. Yeah, of course. The aggregates or all those things are formed, so they contain the disordered regions. Yeah, yeah, functionality, right. Disordered region is not interfering with the functionality. Disordered regions are allowing the protein to take different segments. There are multiple structures which it can adopt, which is possible through that intrinsically disordered regions. So that means they are not fixed. They can adopt different types of such structures, due to which they are bringing flexibility in the protein structure. So that's why they are very much means they can bring flexibility in the protein structure. So, for example, alpha helices, they are fixed. That this is the structure of alpha helix. But those intrinsically disordered regions, they are because they're not having a fixed structure. There is possibility of multiple conformations or structures it can take. So they are bringing flexibility to the structure. So in the next slide, in the next session, I'll include the disordered region also, and we'll understand that how they are responsible in different processes in our cell. Thank you. So I'm ending the class. Yeah. So are you not able to get these slides? These are already shared in the sheet which was provided to us. So maybe in the I guess in your NPTEL course, in the bottom, you can see the option of problem solving session. So whenever you click on that, they will give you some link to click in which you will be able to access the Google Sheets. In those Google Sheets, I'm providing you the link of the Google Drive in which all the presentations will be present. And along with that, because these sessions are also recorded, So you will be able to find these sessions YouTube links in that sheet as well. Yeah, and if in the next session, if you are not able to find it, then let me know, because in the chat box also I'll share the drive link. Maybe if you are not able to find it, then let me know. Okay. Yeah, thank you. Then I'll end the class. See you in the next session.